welcome back guys. In this video lecture we will talk about the regulation of TCA cycle, or you can say the Krebs cycles. So we will divide our topic into two parts. In the first part we will discuss that why the regulation of TCA cycle it is necessary or important. In the second part we will discuss that how the regulation of TCA cycle occurs. Now come to the first part of our lecture, that why the regulation of TCA cycle it is occurs. So here are two important reasons that's why the regulation of the TCA cycle occurs. As we know that this TCA cycle it is actually a metabolic pathway. So for any metabolic pathway to be regulated, it must have two reasons. The first one, that is to keep the cell in stable steady state. So this is the first reason for the metabolic pathway to be regulated, to keep the cell in stable state. The second one, to avoid wasteful overproduction. Now the second important reason for the metabolic pathway to be regulated, that it must have to avoid the overproduction of waste products. So this is actually the two reasons that why the TCA cycle is to be regulated, or any metabolic pathway to be regulated. Now the second part of our lecture, that how the regulation of TCA cycle occurs. So mainly the regulation of TCA cycle it is occur either by the enzyme or by the level of ADP. So these are the two important substances that will able to regulate the TCA cycle. The first one that is are enzymes, and the second one that is ADP. So these two are responsible to regulate the TCA cycle. So you should have to remember that the TCA cycle it will never stop. This enzyme and level of ADP that will able to slow down the rate of the TCA cycles, although there is no stoppage of the TCA cycle occurs. So mainly three enzymes that are important to regulate the TCA cycle. The first one that is citrate synthase, the second one that is isocitrate dehydrogenase enzyme, and the third one that is alpha ketoglutarate. The first one that is citrate synthase enzyme, the second one that is isocitrate dehydrogenase enzyme, isocitrate dehydrogenase enzyme, and the third one that is alpha ketoglutarate dehydrogenase enzyme, alpha ketoglutarate dehydrogenase enzyme. So these three enzymes that are responsible to regulate the TCA cycles. Now we will discuss each of the enzyme and details. The first one that is citrate synthase enzyme. So look at see here what is the function of this citrate synthase enzyme. If we look to the TCA cycle, so the first initial substance for the TCA cycle, that is acetyl-CoA, is mainly a 2-carbon compounds. So what's the function of citrate synthase is? So citrate synthase mainly it will combine this 2-carbon compound into the 4-carbon compound, that is oxaloacetic acids. So this citrate synthase it will combine acetyl coenzyme A into the oxaloacetic acid and will finally form the 6-carbon compound, that is citrates. So citrate it is mainly the initial product of the TCA cycles. So that's why the TCA cycle it will also known as citric acid cycle, because of this initial products. Now what are the factors or the substance that will inhibit the activity of citrate synthase enzymes? So we have the ATP, NADH, and long-chain fatty acyl coenzyme A. These are actually the substance that will inhibit the activity of the citrate synthase. So these ATP, NADH, and long-chain fatty acyl coenzyme A, these will slow down the activity of citrate synthase, due to which no formation of the citrate occurs. So actually the main purpose of the TCA cycle is to produce ATP and reduce coenzyme. So actually when these ATP and reduced coenzyme that are sufficient NSL, so there is no need of TCA cycle to occur further. So that's why these substances actually inhibit the activity of citrate synthase. Now the second enzyme that regulate the TCA cycle, that is isocitrate dehydrogenase enzyme. So first of all we will see here this enzyme act on which part of the TCA cycles. So mainly when the isocitrate it is converted into oxalosuccinate. So here this conversion it is done through the help of isocitrate dehydrogenase enzyme. So this enzyme it will able to convert the isocitrate into oxalosuccinates. So the inhibitor in this case for the isocitrate dehydrogenase enzyme, these are actually the ATP and NADH. These are actually the inhibitor which stop the activity of the isocitrate dehydrogenase enzyme, while the ADP it will activate the activity of the isocitrate dehydrogenase enzyme. It is actually the fourth step of the citric acid cycles. So again these are actually energy sufficient molecules. So when the cell is sufficient with the ATP and reduced coenzyme, so there is no need of the TCA cycles. So here these substances actually inhibit the activity of isocitrate dehydrogenase enzyme and will slow down the activity of this TCA cycles. Now the third enzyme that will regulate the TCA cycle, that is alpha ketoglutarate dehydrogenase enzyme. So again this enzyme it will play important role in the TCA cycle. If we look to the TCA cycle, so this enzyme it will able to convert the alpha ketoglutarate, one of the intermediate of the TCA cycles. So this alpha ketoglutarate dehydrogenase enzyme it will act on the alpha ketoglutarate and will convert this intermediate of the TCA cycle into the succinyl coenzyme A. So this is actually the function of this alpha ketoglutarate dehydrogenase enzymes. So mainly the inhibitors of this alpha ketoglutarate dehydrogenase enzyme, that is NADH, again NADH, and succinyl coenzyme A. This is actually the product formed from the alpha ketoglutarate by the alpha ketoglutarate dehydrogenase enzymes. So these substances NADH and succinyl coenzyme A it will slow down the activity of alpha ketoglutarate dehydrogenase enzymes. Now another reason to regulate the TCA cycle, that is availability of ADP, adenosine dinucleotide phosphates. So when the level of this ADP it will able to regulate the TCA cycles. Now look at see here, this is actually the inner membrane of the mitochondria. So here these line indicate these are the complex present in the inner mitochondrial membranes. So when this ADP it is available, so normally TCA cycle it will proceeds. So actually this ADP when it is sufficient, so no oxidation of this reduced coenzyme A occurs. Now this is actually the inner mitochondrial membrane. Here the electron transport chain occurs. So this reduced coenzyme, that is NADH and FADH2, these are actually the reduced coenzyme. So these are actually formed in the TCA cycles. So when the oxidation or breakdown of this reduced coenzyme A occur, it will form the oxidized NAD and FAD along with the hydrogen and electron are removed here. So these electron it will take by this complex of the inner mitochondrial membrane. So these electron it will move and the inner mitochondrial membrane from one complex into the other complex. So here these proton that are formed from the mitochondrial matrix into the inner mitochondrial space. So here the hydrogen it come here. When this hydrogen it will pass through this ATPase complex, it will form this ATP. So when this amount of this ADP it will less. So here the accumulation, no oxidation of this NADH2 and FADH2, no oxidation occur through the electron transport chain. So here the accumulation of these coenzyme occur in the mitochondrial matrix. So it will ultimately not form the NAD and FAD, which is very essential for the TCA cycle to occur. So here it will slow down the activity of the TCA cycles. So this is about how the regulation of the TCA cycle occur. Mainly three enzymes that will regulate the TCA cycle, that is citrate synthase, the second one that is isocitrate dehydrogenase enzyme, the third one that is alpha ketoglutarate dehydrogenase enzyme, while the third one it will depend on the availability of the level of ADP. So these are actually the how the TCA cycle it is regulated. Thank you so much.